session. Today we have José Paulo Silva. He's a researcher at CCMAR and at the moment is coordinating the structural chemistry laboratories. And today he's going to give us an overview to the main methods on microplastics and nanoplastics. So I'm going to ask everyone to keep yourselves or your microphones muted throughout the presentation. So please, José Paulo, when you can share your presentation with us.
Okay, thank you, Anna. I'm going to start sharing. Okay, now from beginning. Okay, can you see my computer, the title of my presentation?
Yeah, everything okay.
Okay, thank you. So thank you for your words, Anna, and thank you all to attend this seminar. The seminar is entitled Size Matters, Looking for Invisible Plastics, which is also the title of a project we are working on. So the outline of this seminar, first, I will give a general overview of the available analytical methods of microplastics, not exhaustive one, but the main methods will be discussed. Then I will specifically speak about spectroscopy methods. In more detail, the infrared microscopy. CCMAR has one infrared microscope, and we have been working with it in the analysis of microplastics. Then I will move to mass spectrometry methods, both LCMS and GCMS methods, and finally fluorescence methods that we have been using and published some results recently. So the goal of the project is to set up an analytical workflow to identify and quantify nano and microplastics in water, salt, sand, and marine organisms in order to support the development of plastic-free blue biotechnology processes and products. And why this analytical workflow is because there is no single method that can be used to analyze microplastics. Usually we need different methods to get the information we are looking for. Okay, here there's a pictorial view of our project. Here we have a magnifier, you have a plastic bag, breaking down into small pieces, the microplastics. And then we have the spectroscopy methods with a picture here of our infrared microscope. We have the mass spectrometry methods with GCMS and LCMS. And we have the fluorescence methods here with the scheme of our probe, or the probe we have been using, that is pyrene. So, but before going deep into these available methods, we have to discuss the separation methods, that is the sample preparation procedures, which are a key to get a good analysis of microplastics in samples. And in general, there are three main sample preparation procedures. One is density separation. Another one is digestion of sample. We are talking about samples from marine origin. Digestion is very important when the samples have natural organic matter or living organisms, and also filtration to help to, which are a first help in order to select the microplastics by size. The analysis methods can be grouped essentially in two. The first group is spectroscopy methods, where intact particles are analyzed using FTIR or Raman. The other group is mass spectrometry methods, The main machines available for this are GCMS and LCMS. We have to be aware that to use GCMS and LCMS, we have to break our microplastics into smaller pieces, that is molecules, so that we can use techniques that analyze molecules, GCMS and LCMS. There is also fluorescence, but fluorescence needs external compound that is a probe to detect the microplastics. Let's start then with the infrared microscopy. This is a picture of our infrared microscope. It is quite new, as was installed near 1 year ago. It has 3 detectors. Detector that is called DTGS that works at room temperature. And it has also 2 detectors that need cooling with liquid nitrogen. One is the MCTA single element, and the other one is MCT array. And we have 3 different ways of collecting information from our sample. So we can combine the 3 detectors with these 3 ways of collecting information. One is reflection, the other one is transmission, and the third one is attenuated total reflectance, called ATR. I'm going to start with this last one. The ATR is this piece here you are seeing. It has about 15 centimeters long, and it's into the instrument in this way. I'm not sure if this is because ATR gives us the best infrared spectra. This is a spectrum of polyethylene, very good spectrum obtained with this way of collecting information from the sample. Here you have the CH stretch, here you have the CH2 bands. So this is typical of polyethylene. And we also have this tiny peak here that is telling us that this polyethylene is a lower density polyethylene. Sorry, someone was phoning. Okay, this, well, is a spectrum of polyethylene. But we also took a spectrum of this polyethylene after a degradation process. As you can see, the CH stretch decreased in intensity, and there was an increase of this peak here that was assigned to a carbonyl stretch. And also this broadband here that is due to OH stretch. Means that degradation of polyethylene is incorporating oxygen, and that can be seen in the infrared spectrum. Now, another way of sampling is the so-called transmission mode. In this transmission mode, silicon filters are used. They are very small here, about 1 centimeter square. They are nearly transparent to infrared radiation. And therefore, if we had tiny particles on its surface, the system is able to get the infrared spectra with not so high quality as ATR, but with good quality too. As it is a filter, it needs a filtration kit. Here you have the filtration kit, you have a container for water, and you have here Buchner funnel. You have this fitting device. That's the bottom part of the fitting. Here you have the filter where you can put your filter and then a casket, and then you set up and you can filtrate. And at the end, you will have something like this. This is a filter after filtration. You have many particles, including fibers. And if you want to analyze the bottled water, just have to filtrate, for example, 0.5 liters of it, and then the filter is ready for analysis using this system. The software of the instrument analyzes this image here, that is the image of the filter, and then selects the particles and automatically collects the spectra of each particle selected, as well as the corresponding background. And at the end, we'll give a report with the spectra and the number of particles, the identity of particles, the shape of particles, everything we need about the particles that were detected on this filter surface. It can be quite, it can take a long time. That depends on the number of particles we select on the filter. One of the disadvantages of this method is that the method is very sensitive. So if you have samples with many particles, you have to dilute your sample, because otherwise they will overlap with each other and you are not able to quantify the particles nor identify them. Now the other group of methods here, that's the LCMS and GCMS. Let's start by the LCMS. I will not show results, our results from LCMS. This probably can be a second seminar. I will present some results of others and explain what is beneath the use of LCMS to analyze microplastics. This is a paper in 2013 by Wang and collaborators. Basically, they took this polymer, polycarbonate, they make a chemical reaction, hydrolysis, and this compound resulted from the depolymerization. Another polymer they studied it is this one, that is PET, and then the released compound was this one here. And then they use HPLC to quantify the polymers, but by quantifying the compounds that resulted from the breakdown of the polymers. Another paper from Pang of 2020, they basically follow the same procedure. That is a type of nylon that was breakdown using acid in this case, and then the compound was analyzed by LCMS. So to analyze polymers by mass is to break and the particles, the microplastics, into even smaller pieces, the molecules, so that you can analyze them by LCMS and also by GCMS. You can see next slide. So general, the idea is you have to make a reaction and analyze the building blocks of the polymer. So if we, but there are some polymers like polyethylene and polystyrene that you are not able to resulting molecules. You need to use another reaction, and the reaction is called pyrolysis. And with pyrolysis, you can basically heat your polymer, you can break down your polymer into volatile molecules, and then you can analyze them by GCMS. Essentially, there are two types of GCMS that can do this. One is pyrolysis GCMS, and the other one is thermal extraction desorption GCMS. Both ways of detecting microplastics, and both GC need a special equipment connected with it that is a pyrolyzer and a device for thermal extraction desorption. So we don't have any of those specific ways of collecting volatile molecules from polymers at CCMAR. So we developed a manual one that is a scheme here, a very simplified scheme. It's not the complete system, but basically it has a container with, made of quartz where you can add your sample, you can eat your sample, and then the compounds are released up to this larger container. They can be collected using a solid phase micro extraction fiber and the syringe, and then they can be injected into the GCMS. we designed one of those systems, and a glass blower constructed it. The result of GCMS is always a chromatogram, like the one here I'm showing you. Each of these peaks corresponds to a single compound. You have a monitoring of the intensity of all ions that are being detected by the mass spectrometer over time, and this is called chromatogram. Let's see more specifically one example obtained by us recently. This is the same chromatogram here. We have these peaks, you have time, and you have on the epsilon scale, you have the total ion current that is arriving into the detector. The interesting aspect of this kind of information is that you can get a mass spectrum for each of these peaks. Here, you have the mass spectrum of the peak that came out at 5.92 minutes. And with this mass spectrum, you can search in spectral libraries and you can identify the different peaks, okay? So the first peak was styrene. So it looks like that this is a spectrum or a GC profile resulting from the thermal degradation of polystyrene. So the next peak has this spectrum and was assigned to benzaldehyde. The other one was assigned to acetophenone. And the fourth one was assigned to indenone. So with this information, we assign all of these compounds using the mass spectra, and all of these products match with the thermal degradation of polystyrene. And in fact, this is the product. This is a distribution of products that resulted from the pyrolysis of a standard of polystyrene. Very interesting is here that you have compounds with oxygen here, and oxygen also here. You have 3 oxygens. So the products that are being formed include oxygen. And the reason for this is because the pyrolysis was performed in the presence of oxygen. So if we remove the oxygen and would make exactly the same thing, instead of benzaldehyde, we would have only styrene. Instead of acetophenone, we would have this ethylbenzene. And instead of this compound here, we would obtain it, but without oxygen. So it is very important that we control the reaction, the thermal degradation of the polymer, in order to arrive to compounds that we can use to identify the polymers. So the nature of the compounds detector indicate what a polymer is in your sample. And you can use any of those to make a calibration curve and quantify the amount, in this case, of polystyrene in your sample. Let us see one example here. This is a chromatogram resulting from the pyrolysis of a sample of sand. So as you see here, it is quite complex, okay? But because we are using mass spectrometry, we can, and you have, for example, here, the mass spectrum of acetophenone. We can seek, for example, for this ion at M/Z 120. And if we do this, we will get this chromatogram here in red. And you see this peak here at 8.83. So this sand sample contained polystyrene. Okay, now moving to the fluorescence part. The fluorescence, how does fluorescence work? To the analysis of microplastics, usually you choose a probe, a fluorescent probe, and fluorescent and lipophilic probe. And you add it to your sample, which is mostly in a polar environment. But the microplastics are relatively non-polar, and so your probe will migrate to your microplastic. And if you shine light in your microplastic that has absorbed your probe, you will see fluorescence from the probe, specifically from the microplastic. Okay, that's the principle that is beneath the use of a fluorescence method to detect microplastics. So in a recent study, we choose 3 types of polymers. We choose polystyrene, we choose polystyrene, but with covalently linked Nile red, and we choose carboxylated polystyrene. You will see in next slides why I choose exactly these 3 types of polystyrene. In probes, I choose Nile red and the pyrene. Okay, so this is the system that we have been using for fluorescence studies. It's a system based in George Martin's laboratory. It can be used to get fluorescence spectra both in transmission mode and also in front face geometry. Transmission mode is for solution. The excitation beam comes at 900 degrees from the detector that collects the fluorescence. But for solids, the light doesn't penetrate in your sample, so you have to use a different approach. You have to use a cell, and the light goes into the cell, reaches the cell, and then the emission of light is collected with a different angle to avoid the scatter radiation. But okay, this is quite trivial way to get a fluorescence spectrum on solid surfaces. So, but before going deep into the fluorescence results we got, I would like to explain a little bit of the basic concepts beneath the fluorescence phenomena. And basically it is called Jablonsky diagram, which is a diagram showing the energy levels, the electronic energy levels of molecules and the transitions between them. Here you have the ground state, the state of less energy of a given molecule, but the molecule also has other energy levels, they're called excited states. If you irradiate the molecule at one wavelength that the molecule absorbs, you will populate these excited states, and that takes place in the femtosecond time scale. So usually there is a vibrational relaxation, which is a relatively fast process too. And then other phenomena take place from this level here, that is the low vibration energy level of the excited state. If it is a radiative transition, that is a transition from the excited state to the ground state with emission of light, this process is called fluorescence. There are other processes, non-radiative, taking place more or less in the same time scale as fluorescence. They take place in the nanosecond time scale. And also there are other competing processes like intersystem crossing that leads to other states and leads to other processes. With this, what I want to show is that fluorescence takes place from this excited state, but there are other processes competing with fluorescence to deactivate this excited state. And that is one of the main reasons why very few molecules are fluorescent. Pyrene and Nile red, they are fluorescent because the excited state lives long enough to release its excess of energy in the form of light. There are also some note I want to make here. The excited state can also react. And so if the molecule reacts, it doesn't lead to any fluorescence. or can be quenched by other compounds that are nearby, namely oxygen. So oxygen has a strong influence in fluorescence behavior. It decreases the emission intensity. Now let's see specifically Nile red, and the major information you need about Nile red as a fluorescent probe are in these two references, both references from the 80s. So this is a relatively old procedure of using these fluorescent probes to study polarity, the environment polarity. What these references tell is that the intensity emission depends on the polarity of the environment. Here you can see that in a non-polar solvent like an heptane, you have the emission intensity is about 3 orders of magnitude larger than in a polar environment that is ethanol. These references, so this means that when this compound migrates into microplastics, it will increase the emission intensity. Another information about Nile red is that there is the maximum emission changes depending on the polarity of the environment. In this case, in benzene is 575, while in methanol is 642. So this is our spectrum we obtained on silica surface. Silica is a surface similar to sand, so it is relatively polar. Nile red has a maximum emission at 644 nanometers. On salt surface, we got a low intensity emission, but you should be compared with this, the two time scales, the two emission intensities, they are in different scales. But anyway, the emission on salt is quite small. But if we analyze for comparison purposes the emission from the particles of polystyrene with covalently linked Nile red, we will see an emission of Nile red compatible with a non-polar environment, which was expected as, well, Nile red is covalently linked to the polystyrene surface. And if we add to the surface of salt polystyrene and Nile red in relatively large concentration, you will see basically the same spectrum as the covalently linked polystyrene Nile red, meaning that the polystyrene migrated into the Nile red migrated into the polystyrene surface, and that's the reason for this emission intensity that increase, and also this shift in the emission maximum. Let's now change to pyrene emission. The information we need for pyrene emission is in a paper again from the 80s by the Vinick group from Canada. This is the emission spectrum of pyrene in water. And these are two peaks of the vibrational structure of pyrene. We have here I1 and you have here I3, and the ratio of this emission, of the emission of these two peaks depend on the polarity of the environment where the molecule is. In water, for example, the ratio I1, I3 is 1.87, whereas in toluene is about 1, and in hexane is 0.58. So this means that the ratio of these two emission bands can be used to figure out what is the polarity of the environment where the pyrene is. So for pyrene on salt, what we got for pyrene alone on salt surface was this emission spectrum, which is very weak and very noisy. But if you had polystyrene with the same concentration of pyrene, you will get one emission. You will see here I1 and I3 more or less of the same size, meaning that pyrene is sensing a non-polar environment similar to toluene or to benzene, which is actually the environment offered by polystyrene. We were wondering if we could use this emission increase to quantify the amount of polystyrene on salt surface. And what we got is this graphic here. You have an increase of the emission with the concentration of polystyrene on salt surface. If we go into smaller concentrations, here is in the range 0.20, but the behavior is also linear in the range between 0 and 1.25. So this is salt, pure salt, and ACl with pure polystyrene. What happens for natural samples of salt? For natural samples of salt, because we don't know exactly what is in the salt, to quantify microplastics, hydrophobic particles, I would say that's better to say hydrophobic particles on salt, we follow one analytical method called standard addition method. That is taking into account for the matrix effects on the analysis of a given material or compound in a given matrix. So basically we took a sample of salt, natural sample, and we had different amounts of polystyrene, and we got a curve that we extend up to the negative part of the axis, x-axis, and then with this number here, the negative, we can calculate the concentration of hydrophobic particles that are on the salt surface. This is called the standard addition method. Another thing we were wondering is if pyrene is able to enter into a polystyrene and is also able to bind to polystyrene that was transformed. So we studied carboxylated polystyrene as a kind of probe to verify if in nature, where many of polystyrene and also microplastic particles can be degraded a little bit, if pyrene is still a good sensor. And what we got was, this is the spectrum of polystyrene, pure polystyrene, with the two bands here, I1 and I3, and then carboxylated polystyrene, and gives essentially the same or a similar spectrum, meaning that this sensor, this probe compound, fluorescence sensor, is able to also detect degraded particles of microplastics. Okay, so the team that was involved in this work as a CCMAR team includes Alexandra, Camila, Inês, Joana, and José Paulo, and also the team from Necton, because we have been analyzing several samples of salt coming from Necton, SA. And I also have the support on this fluorescence and photochemistry part of these probes, supported by Professor Luís Felipe Ferreira from Technical University of Lisbon, and these collaborators from US, Ramamurti and Stefan. And I want to thank to Fundo Azul that is basically funded our research project. Okay, and thank you all for your attention. And if you have a question or doubts, please go ahead.
Thank you very much, José Paulo. So we move on to questions. So if anyone has it, so please you can unmute yourselves and ask directly to our speaker.
José Paulo, I have a question. You spoke about the use of pyrolysis and thermal degradation coupled to GCMS. But I think it would be difficult to distinguish among, for instance, natural degradation and the ion degradation that is provoked by these methods, by pyrolysis or by thermal degradation. Can you say something about this?
Yes, sure. That is absolutely correct. If you analyze a natural sample directly without any extraction or separation of the microplastics, namely styrene itself can be released after pyrolysis of organic materials. So it might not be a very selective compound, but there are others. During pyrolysis, other compounds are released, namely dimers of styrene, as well as trimers. And these, they show up in very relatively low concentration, but these you can definitely use to identify polystyrene, definitely, because I'm not seeing any reaction involving natural materials that might give rise to a trimer of styrene.
Okay, I think that means that it also depends on the type of microplastics that we are using to study.
Yes, it depends on the type of microplastic. And also, for example, for PVC, I think as far as I've been seeing in literature, there is no good compound to identify PVC in a pyrolyzed sample. It depends really on the compound.
Yes. Okay, thank you. Thank you very much.
Okay. Good afternoon, all. Well, Zé Paulo, nice talk. I just want to add some details about pyrene, because the harm effect, this vibronic dependence of these hydrocarbons in polarity of solvents, the third vibronic band is practically insensitive to polarity. That's why we make the ratio I1, I3. So I3 is a calibrated band. And that's why this measurement is quantitative in what respects the measurement of polarity. And that's the superiority over other fluorescent probes, is that this ratio is a quantitative measure of polarity. And if we separate molecules or solvents by classes, they all give a straight line independent. The slope will vary slightly, but in all the classes, polar, acids, everything, and that's the paper from Dong and Winnick. They quantitate these dependence varying the nature of solvents. And that's why pyrene is so good to measure this kind of phenomena. Okay, thank you.
Yeah, I totally agree with you. Pyrene is a fascinating probe. I think some people don't like it because pyrene is also a polycyclic aromatic hydrocarbon, and well, is also a contaminant of the environment, and usually people don't like it. But we use it and we treat the residues as we should. And there is nothing wrong in using pyrene as a fluorescent probe. And as you say, it's absolutely amazing. We tried several. We tried the coumarins, we tried others, many others in different, including Nile red, but pyrene is definitely the best one. Okay, thank you.
José Paulo, may I ask you something?
Yeah, sure. Go ahead.
Okay. first of all, just to acknowledge such a nice lecture, very didactic. Thank you very much. I'm just wondering how problematic, you now mentioned contaminants in a different way, but how contaminants can influence your data. I mean, regarding their presence in microplastics, if there is any kind of pre-treatment that you do in your samples to remove, let's say, fat or stuff like that, which may induce, I mean, absorption of these probes if they are not properly clean somehow. Is there any kind of approach that you do to remove these contaminants from your microplastics before analysis? Thanks.
Yeah, that's the removal of mainly other particles from natural samples is really a nightmare. Okay, so it's not really the presence of other compounds that might bind to the microplastic, because if you are using the infrared microscope, you can get a spectrum of your particle, and you can get, well, a combination of, you can search for a combination of spectra from the libraries that give rise to the spectrum you are looking for, the experimental one. So you can play with that, but the problem is really the particles. And using the infrared, other particles is using the infrared microscope with these silicon filters is really, really complex, because from biological materials like organisms or so, it's so difficult to remove everything except the microplastics that I don't have a method that I am full happy with to process samples containing large amount of organic matter. Okay. That's what I can say about
I can imagine that it's quite painful with those complex samples. Okay, yes. Thank you very much.
Yes, and another thing is that, when you process the sample, from a natural sample, you always get particles from the environment. So it is also very difficult to process the sample in a very clean way so that you don't get your sample contaminated with fibers that are flying everywhere. That's another problem related. Okay, thank you.
We have a question in the chat. Solange Magalhães asked what type of salt you use.
I used sodium chloride from Pure One, and I used salt flour, and salt prepared in the traditional way that was gave us by Necton company.
Thank you. Does anyone have more questions for José Paulo? Okay, if not, we can close the session for today. So José Paulo, thank you again for your presentation. And we see everyone in the next CCMAR seminar. So thank you, everyone.
Okay. Thank you for your attention. Bye.
Bye, see you, thank you.
Obrigada.